two programs, but several programs in parallel. I mean, right now we have 7 clinical trials going. We'll pretty soon have double-digit clinical trials, lots of catalysts, lots of data coming, and we're learning from all of these to further inform our programs, like our immuno-oncology programs. So we learn from data of 200-plus patients to inform what we want in our next-gen programs, and we're putting that into play. So overall, we feel, I always think about the company in terms of performance, but also health. We feel like we're very good in terms of performing against our assets, but we also built a strong company that could be sustainable and be the leader for a long time to come.
Gotcha. And Sam, can you talk a little bit about the regulatory backdrop? That's always a point of a lot of anxiety, when you have some companies, going on either clinical hold or, some surprise regulatory decisions. Where do you think, kind of FDA is today with comfort and editing as a platform?
Yeah, we've found the FDA, but also the European Medicines Agency and other agencies like MHRA to be very supportive around gene editing. The mistake that I think a lot of the investors today are making is overlaying patterns from viral-based therapies onto gene editing. If you look at the 2018 guidance, that document that came out from the FDA, one of the things that's most striking in that document is the notion of variability. The agency doesn't like a lot of variability in the drug product that's been produced. When you use viruses, it's no pun intended, invariably the case that you have a lot of variability because of the biological nature of how viruses work. And so, patterns that you see on TechOps or CMC from an AAV-based therapy or a lentivirus-based therapy doesn't necessarily apply to a gene editing-based therapy. What we have is a much more deterministic mechanism of action with CRISPR-Cas9, where every time we manufacture, we get very consistent editing, very consistent drug product, and that gives the regulators a lot of confidence, but comfort around the fact that every patient is going to get the same type of therapy. And this is something that, we also had the benefit from the early going with Vertex of saying, let's design a process that's ready for commercial right from the get-go, versus taking an academic process and translating it or comparing it or modifying it to fit commercial purposes. And so all those give us, significant advantage in terms of those regulatory discussions. And I think what we've seen from the agencies, I should say, is nothing but support to bring a medicine like CTX001 or exa-cel to patients that have no other options, especially, in the sickle cell space with the failure of the confirmatory trial for crizanlizumab. There really is nothing. And so, this could be a major boon for these patients suffering from this terrible disease.
Well, let's talk about exa-cel for a minute. So, when you look at your clinical, program, the package, has, years of exposure and very consistent, risk-benefit profile. Give us a perspective on, kind of what you learned as you were interacting with regulatory agencies in U.S. and Europe about, what requirements they're looking for and maybe how that informs your development of other edited products.
Yeah, I think, a key to developing these cell-based programs or cell therapies with editing is getting the CMC right. I think I've seen a lot of companies say, oh, the FDA said this, but we'll figure it out later on. I think that's something we can show comparability later on. And the problem is when you do that, it all of a sudden throws into question all the data you've generated with the process you had before. Can you include that in the pivotal data or not? And I think getting the CMC right is one of the most critical factors and almost a threshold requirement for success with gene-edited cell therapies, maybe slightly different with in vivo programs. And then beyond that, then you do need to get agreement on what the trial design is and how you want to run the pivotal arms. I think right now there are a lot of questions in certain disease areas where, in the past you could do single-arm trials, and now you may need to do a single-arm trial, but also an RCT in parallel, or go straight to an RCT. And that's all very important to get agreement on beforehand. Take an example, our CTX130 program targeting CD70 and T-cell malignancies. This is a patient population that have no options. This is where indications like CTCL, where cancers develop on your skin. One of our patients that self-identified and was the subject of an NPR article is a former NBA player who couldn't get up out of the wheelchair because their feet were full of cancer cells. And then within a month of getting CTX130, this person was walking around normally. And now, in fact, they said they want to travel to Europe for the first time, get on plane. And so that's a transformative effect on this patient. But even in indications like that, I think it's very important, and that's why we haven't guided to when we're going to have a BLA or what the plan is for a pivotal trial, because we want to get alignment before we say here's how we're guiding to not just additional data, but here's how we're guiding to a potential BLA filing. And I think the exa-cel experience has taught us a lot on regulatory and should give us a competitive advantage as we advance more programs, not just into the clinic, but all the way through approval.
And you're right. I mean, there are regulatory bodies, for example, the EMA typically likes head-to-head, and I think U.S. FDA will accept a single-arm study. Do they look at the programs in the context of the technology, or is it the disease itself and kind of the unmet need and the traditional kind of way?
Yeah, I would say the technology becomes a threshold matter.
Right.
There's certain requirements for a new technology, and if you meet those requirements, then it's the standard way of looking at risk benefit. Then they look at the disease, the unmet need, the data that you have generated. And, so far, for the first 3 programs that we put out there, we have RMAT designation for all 3 based on the data. And that tells you that, the agency sees a lot of value in these programs, but they also put that in context of the unmet need that in those indications and specific populations.
Sam, you mentioned manufacturing, so talk about what CRISPR's capacity is, where you see in the next couple of years how many different studies you have the capacity to kind of to get through from proof of concept to even a larger scale phase 3.
Yeah, so I think we've compartmentalized the capacity for autologous separate from allogeneic. And the difference is that with allogeneic, a very small footprint can generate thousands of patient doses, because what you're doing every time you manufacture, you're getting 50 to 100 patient doses, right? And compare that to autologous, where you're getting 1 patient's dose every time you occupy a suite for 7 days, right? And that is a very important distinction that investors need to understand, which plays into gross margins, into profitability, but also how much you need to invest to get to approval. In the autologous context with exa-cel, what we've done is create a modular manufacturing facilities that are redundant, in the sense that we have facilities in the U.S., in Europe, and eventually probably more than that, just to be closer to the sites of care so that we can take the patient cells, get them to the manufacturing facility as quickly as we can, but also be able to supply and modulate how much supply we can manage based on demand without ever having to be in a situation like we had with the Abecma, where there was supply constraint. Right. So we've created that ability together with Vertex. Now, obviously, it's no small investment, but now that we've created that footprint, I think it gives us a lot of flexibility and scalability.
And I think, so Vertex, your commercial partner for exa-cel, has talked about 75 sites for manufacturing. Is that the starting point? Do you think that needs to double over time? And is that capability leverageable when you look to other autologous kind of therapies?
Yeah, so the 75 sites they're talking about are the qualified treatment centers.
Right.
So manufacturing-wise, it's just a handful of sites, U.S. and Europe. But the qualified treatment centers are the centers that are able to do a transplant procedure, but they're also trained in, collecting the cells from the patient, sending the cells to our manufacturing facility, receiving the frozen cells, thawing them properly, and then dosing the patient, right? A lot of this reminds me actually of the medical device space and the launches there. You had these advanced products like TVARs or others that, where you needed like the cath lab, you needed qualified imaging specialists to be able to do these implants. And it took a whole system of training to be able to enable them. So these qualified treatment centers are going to be critical in enabling the launch. And we eventually want to target 75, but, early on, I think even if you get going with, somewhere in the 15 to 20 range, that gives us enough demand in the early going to get the product launched properly. But eventually we want to have 75 centers, but also globally we may expand to beyond that as we reach into patient populations in Asia and Africa. I think there's several, there's 100-fold more sickle cases in South Asia versus the U.S., for instance. Now, the affordability is much lower, and a very small sliver of those patients may be able to afford it, but even that small sliver is a meaningful number. So I think eventually we do need to get beyond the U.S. and Europe, and there's significant unmet need and demand outside.
And Vertex, your partner, has talked about the pre-commercial kind of considerations when you think about, the pricing, reimbursement, access. So to what input does CRISPR have in those conversations? I know it's more than just a handoff. I mean, there's an interaction there, but I wasn't sure the degree that you guys help, with the commercial launch.
Yeah, I think we're doing everything we can to help Vertex. I think, we're lucky to have a partner like Vertex that has tremendous experience in launching in rare diseases. They also have the built-in infrastructure, both the U.S. and Europe, to leverage relationships, for instance, with payers. And I think that's going to give us a very big leg up versus others that are launching the same space. I think to the extent that, there's strategy around how we think about patient populations, how we think about demand, all that, we're here to support Vertex. I think right now we're mostly busy with helping around CMC aspects to make sure that we get over the finish line on the BLA, or getting the approval, but that's our focus right now, and we'll be helping Vertex, but they're clearly taking the lead.
Right. And when you think about the sort of next gen, the conditioning regimen, as part of exa-cel, is we call it first gen. I mean, it doesn't, at all like downplay the data itself, but like when you think about kind of next steps of how you improve upon that, walk through, how you imagine the myoablation, improvements, and how can those lessons be learned in future programs for CRISPR?
Yeah, absolutely. I think, one thing I'll say is, even with the exa-cel the way it is right now, the addressable market is very large. You just do the math. Too early for us to comment on pricing, but if you take the ICER pricing of $2 million per patient and 25,000 patients in the U.S., that's a $50 billion addressable market. The key question is, how quickly are you going to penetrate that market? And how are you going to expand beyond the 25,000 patients? And that's where myeloablative, gentler conditioning agent comes in. If you have a gentler conditioning agent where you don't have to go through the whole busulfan transplant process, and you're selectively depleting the CD34 cells in the bone marrow and then you're reinfusing with the edited cells, that'll make it that much easier. And you're going to have two effects. One is more patients are going to raise their hands to get this therapy that have milder disease, but then you're also going to have a better penetration rate within the severe population. Now, it's not that straightforward to bring a gentler conditioning agent to the market. And one of the reasons we've seen failures in the space is most of the companies that were dedicated to developing gentler conditioning agents were developing it for other indications, like an AML or something else, where the PK/PD profile of those agents were optimized for a different need. What Vertex and us are doing we're developing it solely for sickle cell and thalassemia. So what we want is an agent that has, pretty quick half-life so that it disappears and doesn't impair or doesn't affect the drug product that's infused, right? For instance, which may not be the same for AML. So we're optimizing parameters that's bespoke for sickle cell and thalassemia, which gives us greater confidence that we can bring something to bear in these indications or with this therapy, whereas others haven't been able to.
And the beta thalassemia end of things, so maybe just for reference, just give us your perspective on compare and contrast with like the Bluebird kind of platform and where you see kind of the risks there.
Yeah, I think Bluebird, kudos where kudos are due. They've been in the space for a long time, and they were pioneers in developing transplant-based cell therapies. Now, that said, I think given where we are now with the timing being almost very, very close to each other around potential approvals and potentially getting to the market in sickle cell, which is a big market, I do think CRISPR-based therapies have a huge leg up. One, because the narrative around CRISPR, a deterministic approach where you'd have a precise edit to your genome, is something that patients seem to grasp onto much stronger, more so than the physicians. And the physicians themselves also feel like the unknown unknowns that you may have with the virus-based approach is better controlled with the CRISPR-based approach. And I think that's going to play into the choice that the physicians and the patients make together. In many cases, the parents making the decision. And I think you're going to see a significant tailwind for our product relative to others out there. But again, I think this is more about a category buildup versus a one versus another. So I always am careful about what I say of exa-cel versus Bluebird. But my own personal take, without having seen similar competitive programs before, is that the CRISPR narrative is going to capture the imagination of the patients. It's going to give them more comfort, and that's going to lead to a relatively big advantage in market shares.
Yeah, I gotcha. Let's switch gears to the CAR T. So you guys have had some experience in hematology and solid tumors. Just for those in the audience, so give us some perspective on the next gen kind of CAR Ts and how that's different than your first wave of experience.
Yeah, and I would say this is a entirely new paradigm in drug development and discovery. In small molecule world, what you do is you develop a small molecule for a particular indication. Either it works or it doesn't. Sometimes you have a backup small molecule, but it's not a improvement on the first one. It's a backup. Sometimes you have follow-ons that you think may be better. But what we're doing here is to develop take a particular thesis and say cell-based therapies are going to be very important in cancers because they can actually completely eliminate the cancer and lead to cures. And what we're doing is putting the first gens in and then improving upon that. With everything we learn from the first gens, whether it's patient biomarker data, whether it's patient PK/PD data, we're putting into play with the next gens. And with the next gen program that we have at CTX112 and 131, we've been able to cycle it very quickly. Within 18 months of the first gens, we brought the second gens to the clinic now, where we're enrolling. We disclosed yesterday that we're enrolling patients. And we haven't shown that much data on it, but there was a recent publication by Carl June, the person who invented CAR Ts, and had a similar edit, Regnase 1, with a different co-edit called ROPIN1. And that publication showed that these next-gen CAR Ts are not just 10 times better, 100-fold better in terms of expansion, cancer cell killing, and the ability to retain naive state in these CAR Ts, or a more memory-like state. And I think that's all going to come into play with our next gens, which we're enrolling now. Not to mention the advantage we have in terms of manufacturing, because these cells expand a lot more. So every time you do a manufacturing run, you get 100 patients worth of doses. So all that's going to come into play with the next gens. The first gens that we have, CTX110 and 130, I think are products in their own right, and we're going to push that forward all the way. And we're going to bring the next gens forward and decide how we're going to position one versus another over time once we see more data. But at this point, I think a few things have happened. The Carl June paper, but the fact that in the next few months we'll have PK/PD data around the next gens, and all the pharma companies are now picking up the phone saying, "Oh, we want to learn more about your next gens." I think they went dormant for a period of time there before that, but now I think there's a lot more interest and a lot of interest from the investigators, which is why we had the presidential symposium at the EBMT conference last month with our CTX110 data, and the interest is just growing. So while investors may be thinking that cell therapies have hit the nadir, I think the investigators and pharma companies are just ramping up on their interest.
Right. So the next gen could have the potential of longer durability of effect and maybe better responses at the onset?
Both. I think hopefully, we'll have a greater proportion of patients with complete responses or greater responses in the early going, and those responses are deep enough that lead to durability, and so you have a greater proportion of patients reaching, let's say 6-12 months of durable responses.
With the next gen technology, talk a little bit about the safety profile. Is the CRS, and the like, or would you expect there to be tweaks to that as well?
Unknown at this point. We have to see what the data look like, but that is something that we're watching very carefully, just to make sure that the increased cell expansion doesn't lead to more CRS. But CRS is something that investigators will figure out how to manage now. I think if you look at all the advances in how people use TOC, how quickly they intervene with tocilizumab, I think CRS has generally been a managed phenomenon lately.
That's perfect. And when you look at the sort of compare and contrast of a lot of the experts that we've talked to years ago have said, bispecifics aren't with respect to CRS, you have recurring CRS and maybe the efficacy isn't there, but there's been an evolution of bispecifics in some of these liquid tumor types. Give us your background on where you think a CAR T, a cell therapy, would stack up, against some of these next generation bispecifics.
Yeah, it's going to be very fascinating dynamics take the CD19 market, positive lymphomas, for instance. I think what you're going to see, if I take my crystal ball and say what's going to happen in 5 years, I think in the academic medical centers, they're still going to do a lot of autologous therapies because they have access to those therapies, but that's a very small portion of the market, like 15% of the market. The majority of the market, 85% of the market, is in the community settings. And there, I think it's going to be, I don't think it's going to be one winner take all situation, but there's going to be both bispecifics and allogeneic CAR Ts vying for the same market. And allo CAR Ts have a significant advantage in a sense that it's one dose, and a lot of the patients that are treated in these community settings are coming from faraway places. They're not living right next to the centers where they get treated. And bispecifics, where you have to take 9 courses or 10 courses of treatment, and each of them having risk of CRS or even potentially ICANS, I think that's not going to fly in the community settings. I'm not saying that means that bispecifics are no-go. I mean, right now, it's pretty impressive data with the pertuzumab and glofitamab, for instance. There will be penetration. But if allo CAR T reaches its potential, I think you're going to see allo CAR T as the preferred modality in community settings.
Gotcha. And just give us a status update on where we are with the next-gen assets. So 121, 131, the CD70 program.
Yeah, so right now what we have is CTX110, where we're pushing forward, pedal to the metal in a registrational trial, where we hope that the 2-course treatment, the data are better than the 1-course treatment, and that is a very competitive profile that can get approved. CTX130 in CD70, we're continuing to have regular discussions with regulators to understand what is required for a registrational trial. We've shown sufficient data. We showed some data at EHA last year, and we've shown tremendous benefit for patients that have no other options, in these indications. And so we want to, again, to our earlier discussion, want to get complete alignment to say this is what the registrational trial could be, and we're going to push forward on that in T-cell lymphomas. Gotcha. For solid tumors, we're going to move to CTX131. And we're very confident in that because, one, you're seeing other programs targeting CD70 show responses in CD70 positive solid tumors. We had the world's probably the first complete response in a solid tumor with an allo CAR T or any CAR T last year that we disclosed in RCC. And we see a lot of biomarker data in the patients we've treated, which gives us confidence around CD70 as a target in solid tumors. So we're going to push forward with CTX131 in solid tumors. And that's probably our biggest, shot in the portfolio, in our portfolio, where that works. We're a completely different company. And then we're going to move forward CTX112 and CD19 and see how it compares with CTX110. So there's a lot of it's hard for everyone to keep track of everything we're doing, but a lot of things in motion and a lot of cards to turn over. But that's the advantage we have, is that we have more information, more experience in the space than any other player in the space.
And you're right. So with the RCC of the kidney cancer patient, there's been fewer CAR T programs that have had success in solid tumors. I mean, hardly any. But historically, you haven't seen cell therapy for solid tumors, widespread because of T-cell exhaustion and durability questions. So help us with kind of how you guys are thinking about evolving CRISPR to address solid tumors in a more effective way.
Absolutely. And the key consideration in solid tumors is exhaustion. Yep. I think when we looked at all the patients we dosed with CD70 in solid tumors, we took the cell samples out and we looked at these CAR Ts. And the biggest phenomenon we saw that's different from hematologic malignancies is exhaustion comes early in these CAR T. So it's going to need next-gen edits. And that's happening around both autologous and allogeneic cell therapies. At AACR recently, there's some very interesting data on GPC3 targeted auto CAR Ts with the TGF-beta-R2 edit, similar to what we're doing. Yeah. And in fact, most people won't know, we have a GPC3 auto CAR T program that's also moving forward with the same edit. So these next-gen edits are becoming very important. There's a recent data set around expressing cytokines like IL-18 in these auto CAR Ts, and that makes them much less prone to exhaustion in these solid tumors. So the frontier of solid tumors and CAR T is going to get cracked in the next 18 months, and you're going to see a tremendous resurgence in this space. And what's also going to be clear is that traditional lentivirus-based CAR T is not going to work, and you're going to need edited versions that are much more sophisticated to make it work in solid tumors.
Right. And outside of the hem-onc setting, some companies are evolving, CAR Ts to, autoimmune or, sort of I&I type of indications. Like, talk about, with CRISPR, you guys are also looking into rare diseases too. So what are the opportunities outside of the exocell and hem-onc kind of franchises?
Yeah, so I think we have 3 other franchises now. So obviously autoimmune we're looking at, but what we're doing is fundamentally trying to change the manufacturing process where the efficiency of manufacturing is that much greater, because in autoimmune is a much bigger market and may require more than one dose. So we want to make sure that we're not spending, and the price point is different, autoimmune versus cancers. So we want to make sure that we have a profitable product. And so we're trying to reduce the COGS of these products relatively low so we can make an actual product there. But I'm very excited about in vivo, where we're going to dose patients in the near future. We announced yesterday in our Q that we're moving forward multiple in vivo programs, and these are for targets like ANGPTL3, LPA, where the biology is getting de-risked with RNAi. And so single shot therapy where people have reduced their LDL, cholesterol, or triglycerides by 50-60%, that's a pretty meaningful advance for society. I mean, we're going to see a big impact on when people die from these therapies, because you're going to add like 10 years to their lives, given the cardiac disease is the #1 killer that we see. So we're excited about that. Our diabetes program, together with Vertex, is moving forward quite nicely. We're dosing more and more patients there. So we're going to have plenty of cards to turn over in the next 12 months.
Excellent. Well, with that, we're out of time. So, Sam, thank you very much.
Yep. Thank you, Joy.